I'm Mary.
I'm Zhang, and today we'll be talking about DNA structure and replication.
So first you want to talk about DNA structure, and the structure of a DNA is it's a double helix.
And it looks sort of like a spiraling staircase.
Right, and it's composed of nucleotides, and a nucleotide is made up of a phosphate group, a 5-carbon sugar called a deoxyribose, and then also a nitrogenous base. And the nitrogenous bases, they're bonded together by hydrogen bonds in pairs. So adenine always goes with thymine, and guanine always goes with cytosine, and that's part of the base pairing rules. So there are 2 bonds between adenine and thymine, which are shown by the toothpicks right here. And the one toothpick basically just represents the all of the hydrogen bonds. So there's 2 between adenine and thymine, and there's 3 between guanine and cytosine.
We also made these mudding cakes to represent how the nitrogen bases pair together. So here there's adenine and thymine, who are a couple, and cytosine and guanine, who are a couple. And so now I'll be talking about DNA replication, like the actual process. So it begins when helicase, which is an enzyme, goes up the DNA strand, splitting the hydrogen bonds between the nitrogenous bases. Now, these hydrogen bonds are not able to form again because they are blocked by single-stranded DNA binding proteins, which prevents them from
re-annealing.
Yeah, definitely. Also, now there are 2 strands. There's a lagging strand and there's a leading strand. Now, the process of the leading strand is more easy to understand because basically, DNA polymerase III will come and put complementary DNA nitrogenous bases up.
Following the helicase.
Yeah, and that goes from 5' to 3', and it just, follows the helicase.
Right, but on the lagging strand, it still has to move from 5' to 3', but it has to move in the opposite direction.
Yes.
So this is done by an RNA primase.
And this RNA primase puts down temporary RNA primers, and these RNA primers are like mixed on different sections of the strand.
It's and like on this side. And instead of thymine in the RNA primer, since it's part of since it's part of RNA, thymine is actually replaced with uracil, which is shown by the orange jelly bean.
Yeah, so when that happens, and then this acts as a SOS signal, and that signals for the DNA polymerase III to come and replace what sides are not finished with RNA primer with the complementary DNA nucleotide nitrogenous bases. Then DNA polymerase I will come along, and this takes the RNA primer and replaces it with DNA nucleotides. And DNA nitrogenous bases. And so now this whole strand is filled with DNA nitrogenous bases. Now these are just in fragments on the lagging strand.
Called Okazaki fragments.
Yes. Now these fragments have to be connected, which is connected by the DNA ligase that goes along the side and connects all of the Okazaki fragments.
Right. So once you have a continuous strand of DNA on the lagging strand and the leading strand, it creates, as you can see here, two new strands of DNA. And this whole process is called semi-conservative replication. And it's called semi because one of the strands of DNA, such as like just this one right here, is from the old or original DNA, and then the other part is new. So each strand of DNA is identical to each other and to the original strand, with one from the original strand and one that was added by each of the enzymes and proteins.
And DNA replication in prokaryotes differs from DNA replication in eukaryotes, because in prokaryotes it starts from one central point, but they move in opposite directions, encasing the whole circular chromosome.
Kind of like a zipper that moves around a circle, unwinding from both directions and meeting at the end.
Yes, and in DNA replication in eukaryotes, it starts at different origins.
Right.
So thank you.
Thank you.
Yay!